Hello, everyone, and welcome to today's live broadcast, Predictive Biomarkers to Diagnostic Test Scientific and Regulatory Consideration in Development of Companion Diagnostic Tests, presented by our keynote speaker, Janaki Veeraraghavan. She is a biologist at the Office of In Vitro Diagnostics and Radiological Health at the Center for Devices at the U.S. Food and Drug Administration. I am Marjorie Torres of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom or center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credit tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Janaki Veeraraghavan. I will now turn the presentation over to her.
Thank you, Marjorie, and it's a pleasure to talk at the virtual event hosted by LabRoots. As Marjorie described, the talk mainly outlines how you convert a predictive biomarker discovered in research to a companion diagnostic test or any diagnostic test for that matter. But this talk will focus on the regulatory considerations for developing companion diagnostics. I will go through the talk as such is divided into three distinct categories or topics, so to speak. In the first place, I will go through what would be an FDA 101 for most of the audience. We will go over the regulatory pathways and the ways a developer could reach out to the FDA to seek feedback. Since I work with mostly oncology devices and companion diagnostic to date are dominated by the oncology field, and given the interest in immuno-oncology, I'm going to take the examples of some of the IHC tests that was approved by FDA over the last few years and use them as a case study for describing how we come to regulatory decisions. And also important to keep in mind is how and what FDA means by analytical validation. I will spend a significant amount of time talking about that. And towards the end of the talk, I'll go over some of the complexities in how diagnostics are rapidly evolving and what FDA is doing to stay nimble and make sure patients have access to the latest technologies and therapies that are being discovered out in the field. With that, I think one of the first most important things to recognize is the difference between a biomarker versus a diagnostic test. A biomarker is anything that can be measured to indicate a disease state. And back in the day, you could think of this as probably the sugar in urine, and it could also be a test that measures the response to a therapeutic. But biomarkers mostly exist in the exploratory space or in the research space. And given that it is in the exploratory space, the test systems might not be defined. There might be multiple different ways of measuring an analyte or a biomarker, and and it is not very well specified. So there are multiple ways to approach the biomarker. In contrast, a diagnostic test is well defined, and it is intended for diagnosis of disease and to identify appropriate treatment options for patients. And a diagnostic test is also specific in the sense that it defines the entire system from the specimen type, whether you're measuring the analyte in blood in urine or in tissue in the case of oncology, and if that tissue type is fixed in formalin or alcohol, or is it frozen, all of that. So it defines the specimen type, starts with the definition of the specimen types, the reagents that is used, and it also goes over the methodology or the protocol used to arrive at a result, the instruments and the software involved to keep a test running in order to get a result. And diagnostic tests are also ones where we know the performance. We know what the sensitivity and specificity of the test is, or its predictive value, whether getting a test result that's positive predicts a disease state or response to a therapy. So diagnostic tests are extremely well defined, and this will be a kind of a touchstone, which I'll come back to a few times throughout the talk. So how are diagnostic devices regulated by the Food and Drug Administration? It is a risk-based regulation, and there are three classes of risks: class 1, class 2, and class 3. In this slide, I don't point to class 1 devices because they are exempt from FDA oversight, whereas class 2 and class 3 devices fall under our purview. And what is a class 2 device? So class 1 devices are low-risk devices where the risk associated with the wrong result is relatively small, whereas a class 2 device carries a moderate risk. And class 2 devices are then regulated by what is called a traditional 510K pathway. And the 510K pathway gives clearance or marketing clearance for diagnostic tests to be marketed when it shows substantial equivalence to a legally marketed predicate. So a prior test exists, which then the new test can show that it is substantially equivalent to and would obtain an FDA clearance. These are for moderate risk tests. Whereas class 3 devices are high risk tests, and they go through a pathway called the pre-market approval application. The PMA applications are reviewed and approved by the FDA based on demonstration of safety and effectiveness. And I will go over what safety and effectiveness is, or it'll be more clear as we go through the talk. So class 3 devices are high-risk devices, and almost all oncology applications for companion diagnostics fall under this category currently. And a third route called the de novo 510K pathway exists where there are no predicates available. So you have developed a new test and you believe it's a moderate risk device, and it could easily fall under a class 2 where the regulatory burden is somewhat lower than a class 3 device. Then you come in through a petition called the de novo 510K pathway where FDA assesses it for there existing no predicate for it, and the determination of safety and effectiveness as we do for the PMA approval, and then propose a set of special controls and authorize the marketing of the test and classify the analyte into a class 2 if it is appropriate. So these are the three major regulatory pathways, and given that the talk focuses on companion diagnostic, much of what I say today will be applied to the class 3 devices that you see on the right of your screen. So why is companion diagnostics exciting? When you look to the right, you can see blindfolded scientists shooting in the dark, which was the era before targeted therapy and development of targeted therapies existed, where the efficacy was in the overall population was poor. However, when targeted therapy was developed or came into the market as a big pathway to developing oncology drugs, it became apparent that you need that precision medicine or being able to identify people to move to the right of the screen where you're able to target with high effectiveness the patients who are actually likely to respond to drug treatment. It became apparent that you need accurate, reproducible, and clinically useful companion diagnostics to make precision medicine deliver on its promise. So that's what makes companion diagnostics important in the coming era, so that we deliver the drugs to the people who need it and have high efficacy out of it. So the Food and Drug Administration issued a guidance in August of 2014, which defines companion diagnostic as a test that provides information that's essential for the safe and effective use of a corresponding therapeutic. And the guidance document lays out FDA policies for approval and labeling. And I think I would like to note at this point is that all companion diagnostic applications are contemporaneously reviewed as the drug approval goes through. And you will see the implications of that in the next slide. So a companion diagnostic is being developed to identify patients who will respond to a therapeutic, which means the development of the drug, which is pointed to at the bottom of the screen, which starts in the research stage on the left with target selection and drug selection and validation. The device or the test that needs to come in as a companion diagnostic has to be developed in parallel such that early in development, the biomarker that needs to be converted to a diagnostic product is identified. If the biomarker is identified, the best test for segregating responders and non-responders or potential responders and non-responders has to be thought of. So the test at this time could be anything. It could be an analyte in blood, it could be a measure of a protein in the serum, or it could be a DNA mutation. It could be an IHC by identification of a biomarker by IHC in the tissue sections. So at this stage is when the choice of test and a protocol to identify it should be thoroughly researched. And this is followed by a stage where analytical validation of the test needs to be performed. By analytical validation, we need to understand the parameters within which the test performs reproducibly, that it gives out the right exact same results with high precision and reproducible results over multiple testing over time. And I'll come over to the specifics of analytical validation later in my presentation, as I had mentioned. So in the preclinical and early clinical stage, the analytical validation of the device should be undertaken. And finally, once the assay is the technology, the specimen, the platform, and a cut point is identified, this assay now needs to be clinically validated. And this is where the clinical utility of both the drug and device are established in a clinical trial. Previously, it used to happen over 3 phases, and the phase 3 would be a pivotal trial. But in this new era of exciting new drugs coming to market, this might happen earlier in development, such as a phase 2 study. So one needs to be prepared for it. And under the analytical validation arrow, you see three words. It's called, which are pre-sub, IDE, and PMA. I've told you about PMAs. It's the pre-market application that typically comes at the end of the pivotal clinical trial. Before coming to a PMA application, one needs to also look at whether there are earlier points of interaction with the agency. And the earlier points of interaction of the agency could be as early as when you start analytical validation of the test. And this happens to be, this can happen through a process called pre-submission or Q-submission with the Center for Devices. And another important question that is triggered is an investigational device exemption. And the question a developer needs to ask is, since it's a human subject research involved, is an investigational device exemption needed? And an investigational device exemption or an IDE application may be needed if a diagnostic test for a biomarker is used to identify or select patients to a drug trial. And the IDE requirement is based on risks to patients. So does the use of the test and the results from the test pose a significant risk or a non-significant risk to the patient's health? And one can identify whether the investigation in which the device is going to be validated would be a significant risk or a non-significant risk by submitting a pre-submission application to the CDRH. And it's a relatively painless process. It takes about 30 days for us to turn around, or less than 30 days for us to turn around and say if one would need an IDE application or not for a given clinical trial. So I talked about what happens at the end of performing a pivotal trial. And the question that most often comes up is, oh, we have a biomarker. We don't know whether this is going to need a companion diagnostic application. Should we bring the application in? And so the question that is necessary to answer is, is this a biomarker-selected trial? Will an in vitro diagnostic device or a test for the biomarker be required for safe and essential use, safe and effective use of the therapeutic product? If the answer is yes, it probably triggers the need for a companion diagnostic application. But the decision for requirement of a companion diagnostic is a joint decision from drug and device review divisions in CDER and CDRH. So all of these reviews, starting from when the drug application comes in for an IND or an investigational new drug application, or an IDE or investigational device exemption application, all these reviews are conducted contemporaneously between CDER and CDRH, and the review divisions work very closely together to answer these questions. More recently, a new term, a complementary diagnostic, has come into play, and I will touch upon this again later in my talk, but I wanted to lay down here the difference between the two. A companion diagnostic is essential for the safe and effective use of the corresponding therapeutic product as we had previously defined, and I'll be using the examples of PD-L1, IHC22C3 down the road. What is a complementary IVD? A complementary IVD is a newer category of diagnostic devices for a therapeutic product, and this diagnostic device or a test identifies population for which the use of the therapeutic product has a different benefit-risk profile. So the device can identify differential responders, whether it is improved response in positive or increased risk to a side effect. So either a safety risk or increased efficacy in a subgroup of the patient population. And so how does FDA review a companion diagnostic test? Or what are the components of a companion diagnostic test that is reviewed by FDA? So like I told you at the very beginning of my talk, the risk base of the device is based on the intended use and the indications of use. So that is a key component of the device that we look at. Then within that, we expect the application to include device description, the reagents, the instrument platforms that are used, pre-analytical factors that may impact test results, the analytical performance, the clinical performance, the instrumentation and software that are required for generating a test result. We also review the labeling or the package insert for the device. And given that most companion diagnostics are class 3 devices, it also requires quality systems review in the GMP and BIMO for bioresearch monitoring. These are all the components of an FDA review of a companion diagnostic. At this point, I'd like to say that the two key components of a review are the clinical performance and the analytical performance. I will go over clinical performance, which is the most critical component of a device's ability to show safety and effectiveness, and then go over analytical performance requirements that FDA places. So how do you validate a clinical diagnostic test in a clinical trial? So the clinical validation of the device is typically done in the pivotal trial for the therapeutic product, because it's a companion diagnostic. And in this case, the device is used to test patients, specimens, to either stratify or to identify patients who are eligible for the therapeutic clinical trial. And for the analytical validation, these studies have to be done with an eye towards a clinical decision point. We will go over clinical decision point a few slides down, where I'll give you a more greater detailed description of what it will be. So what else does one need to think of when you are beginning analytical validation? I had talked about earlier how a diagnostic device is extremely well defined. The test used to select the target population enrolled into the clinical trial should specify the specimen type that will be used for testing. It should identify the specific test, whether it's a PCR, an IHC, NGS, or what technology is going to be used. It should specify the protocol that goes with the test that will be used, and a clinical decision point. So if it's a quantitative assay, in what ranges will the patient selection apply? If it's a qualitative assay, what is the cutoff at which a patient specimen is deemed positive or negative? This is very important because ultimately we are trying to determine the precision of an assay, and the precision typically around a pre-specified cutoff. Also to keep in mind, before entering a phase 3 pivotal trial, the cutoff should be pre-specified and logged down, and the assay fully logged down. By the assay being fully logged down, we mean all these 4 components are in place and are not modified as the trial goes on. And what will the trial be determining? What it would be, so keeping in mind the intended use population should develop the study design that is used to support the diagnostic claim. And so one also needs to put in place pre-specified clinical and statistical analysis plan, and establish an appropriate endpoint or a surrogate endpoint that will be chosen at which success is to be determined. And typically, analytical validation should precede clinical validation. And what are the choices in terms of clinical trial design for determining whether your test is safe and effective? So since it's a companion diagnostic we are talking about, this can be determined in 2 different types of clinical trials. The first one is a targeted or marker-positive only trial. And anytime a marker-positive only trial is picked up, a companion diagnostic becomes essential for safe and effective use, because that is the only patient population, in which the drug effect has been characterized. Ultimately, the drug trial could also be all-comers, such that all patients in the intent to treat population are eligible irrespective of their biomarker status. And the types of data one derives from the diagnostic clinical trial is determination of the sensitivity and specificity in all-comers trial, and positive predictive value and negative predictive value for therapeutic effect in the biomarker selected population in a targeted clinical trial. So as I spoke before, I will be using immunotherapy approvals as a case study for this presentation. And as many of you would be aware, this is an extremely happening field of clinical research in oncology. And so far, there are 5 different drugs that all target the PD-1, PD-L1 axis that have been approved in more than a handful of indications. And to be clear, this table was not updated recently, and the list of approvals is much longer than the list of approval in different indications is much longer than the ones I have presented here. So, and when you look at this table, all the indications against which there are asterisks present are ones where a companion or a complementary diagnostic has been approved for use in this patient population. And there are in fact more. And here is a comprehensive table that lists all the diagnostics that have been approved in the immunotherapy area by the FDA. We have companion diagnostics for Keytruda which is the Merck drug that targets PD-1 and is paired with the DACO PD-L1 IHC 22C3 pharmDx test. This has been approved as a companion diagnostic in lung as well as gastric cancer. And we have 3 additional tests for identifying PD-L1. In each of these cases, PD-L1 is the target analyte, and we have 3 additional tests, 1 more by DACO and 2 additional ones by Ventana for Opdivo, Tecentriq, and Imfinzi, each of which are used to identify patients with the indications in the second but last column to the right. I would like to draw your attention to the last column on the right, which is titled the clinical cutoff. What you should bear in mind is that each of these assays have a different definition of what constitutes a PD-L1 positive patient or a PD-L1 positive tumor for any given patient. If you look at the top 2 lines, for the same device, the DACO PD-L1 22C3 pharmDx, in the lung indications, there are 2 cutoffs. The greater than 50% tumor proportion score in the first line and greater than 1% tumor proportion score in second line or later. And this identifies patients who are eligible for treatment in the first line or in the second line. And in the case of gastric cancer, it uses an entirely different scoring system called combined proportion score, which includes tumor and immune cells. The tumor proportion scores includes only the tumor cells. So here's an example of the 22C3 pharmDx intended use. It's a very busy slide, and I know it's hard to read. What I'd like you to concentrate on are the boxed items, and the arrows describe what exactly each of these means. An intended use statement, an IU statement, is a comprehensive description of the device, where the test is supposed to be applied, how it's performed, and what does it actually do. So in the first part, you can see that it identifies the analyte. It says it's an IHC test. It flags the technology in the opening. It flags the analyte that's going to be detected. The matrix or the specimen type that is used for the test, and the test system. The Envision Flex is the detection reagent, and the Autostainer Link 48 is the instrument. Reagents and instruments involved in identifying PD-L1. And as you move to which conditions it's used in, it flags that for non-small cell lung cancer, PD-L1 status is defined by tumor proportion score. And in green is the description of how the tumor proportion score is arrived at. And you also flag the cut point for TPS greater than 1 or PD-L1 high, TPS greater than 50, which are the indicators of clinical cutoff. And finally, so if you do this test and run it and take into consideration the clinical cutoff, what are you doing? You are using this test as an aid in identifying lung cancer patients who are eligible for treatment with pembrolizumab. So this is what flags it as a companion diagnostic, and this is the intended use of the test. And you can see there is similar language embedded within the gastric and gastroesophageal junction indication for which this drug approval exists. So the intended use statement gives you a complete picture or a comprehensive picture of how the test is used. So how did one arrive at a companion diagnostic claim? This was based on Keynote trials that Merck conducted. And originally, the Keynote trial was based on objective response rates. I forget the clinical trial name in which it was determined, but objective response rates in PD-L1 patients greater than 50% of tumors expressed PD-L1 was used to support the companion diagnostic claim. And later, The same study updated the data for overall survival in patients that had tumor proportion score of greater than 1. By tumor proportion score, it means all tumor cells that express PD-L1. So using that as a marker, the increase in response, as you can see on the curve on the right here, in second-line patients who had received more than one line of treatment for their non-small cell lung cancer, when compared to docetaxel, had higher survival rates. And this higher survival rate was preserved irrespective of the drug concentration used. The studies did not explore the activity of the drug in patients who were negative or had no PD-L1 expression. That kind of logged into the idea that this is a companion diagnostics, that the efficacy of the drug in a marker-negative population is unknown. Similarly, in overall survival for first line, the clinical study was conducted based on selecting patients that had greater than 50% of the tumor cells expressing PD-L1, and they saw high efficacy in that group when compared to chemotherapy and improved overall survival rates. Since these trials were marker-selected trials, since these were marker-selected trials, this already automatically fell into the companion diagnostic paradigm. I talked about a complementary diagnostic earlier. And given that there were multiple clinical trials with multiple drugs at that time, this is a good example to go over how a complementary is different than a companion. A complementary diagnostic test was approved for nivolumab, and this complementary diagnostic approval it has been for 4 different indications: lung, head and neck, urothelial cancer, as well as melanoma. And if you look at the written portion of the slide, what you can see is that similar to the companion diagnostic, the first part of the intended use statement describes the test and the score, and the second part here in this case is in a tabular format, given that there are 4 indications for which the complementary diagnostic approval exists as of now. You can see that the portions of the text under intended use column that are in red shows that the language is slightly different. Here, the intended use states that PD-L1 expression as detected by the 28-8 pharmDx test in non-small cell lung or squamous cell carcinoma of the head and neck may be associated with enhanced survival. So in this case, it doesn't say that you have to be PD-L1 positive to get the drug. What it says is that if you express PD-L1 positive, the effect of the drug might be associated with enhanced survival. I will talk a little bit more about the melanoma trial, where the intended use statement is correlated with magnitude of treatment effect on progression-free survival from Opdivo. So this was a 3-arm trial where patients who had melanoma and had no prior therapy were enrolled and treated with either nivolumab or nivolumab with ipilimumab or ipilimumab alone. And as you can see on the left, in patients with less than 1% PD-L1 expression, nivolumab was more effective than ipilimumab, but the combination appeared to have more effect. Whereas if you look at PFS in PD-L1 greater than 1, what you see is that both nivolumab and nivolumab in combination with ipilimumab show higher efficacy than ipilimumab alone. And what is more striking is that for patients treated with monotherapy, in less than PD-L1 1% population, nivolumab is less effective than combination, but in greater than 1% population, nivolumab is almost as effective as the combination. That's why we say that there is an increased magnitude of expression in the intended use. And this is the data based on which the approval for the complementary diagnostic was issued. So moving on, having an understanding of what companion and complementary diagnostics are, I'd like to go into some of the factors that impact assessment of or the review of a PMA application. So slide trouble. Okay. Some of the factors that impact the review of a PMA application is the clinical trial assay. So I've kept emphasizing in my talk that a finalized lockdown assay should be used for enrollment of clinical subjects, and the test that's used for enrollment of clinical subjects is called the clinical trials assay. And the market-ready assay is the assay that will be pushed out or supplied to the market. How do these two match up? And what is the definition of the clinical cutoff and the scoring methodology used to determine the biomarker status? We hope that this is logged down in the clinical trial as well. And the exploration of multiple cut points. These are factors that tend to impact how PMA reviews are affected. So what happens if the clinical trial assay is not the same as the market-ready assay, which is the subject of a PMA application? If the clinical trial assay that was used to enroll patients is not identical to the market-ready assay, then a bridging study is required to show the equivalency or the performance being the same for a clinical trial assay and the market-ready assay. So why would this happen? This might happen because more than one version of a clinical trial assay was used to enroll patients because the test itself was still under development as the clinical trial progressed. I have lost my place. Or multiple lab tests might have been used and pre-screening for the biomarker might have been done at local sites such that many different assays were used to enroll clinical trial patients. And mid-trial device changes were made because research information indicated that there was either a problem with the secondary reagent or the cutoff did not appear to be appropriate. And sometimes you have clinical trial assays that have inadequate validation and require major modifications at the end of the trial. And sometimes cut slide stability issues such as that are assay specific for IHCs can bring about differences in clinical trial assay and the market-ready assay. So anticipating these challenges down the road is essential for a successful development of a companion diagnostic. And the best way to do that is to have a robust plan for specimen banking, retrieval, and retesting. So anticipating that you will require retesting of these specimens is a good plan going forward. And how do you do a bridging study? A bridging study will retest all clinical trial positive and a sum of the clinical trial negative if it's a marker selected population with the new or revised test to support safety and efficacy of the therapeutic product. And one should ensure that in a bridging study, the retest population is representative of the intended use population for the device. And since a bridging study is being conducted, one should also have a statistical analysis plan to take into account any discordance from test results from the clinical trial assay to the market-ready assay. And you should also think about samples going missing for the bridging study. Suppose the patient deposited enough specimens only for the clinical trial assay, and there is an inability to retest with the market-ready assay. You have missing samples. And a statistical analysis plan that accounts for all this can give you a robust assessment of equivalency or equivalency of the clinical trial assay and the market-ready assay. And it should also address bias and the impact of any such biases due to missing specimens or discordance on therapeutic product efficacy. So the second point that I raised that might impact cut point for a device is that of a review of a PMA is that the clinical cut point becomes important. The clinical cut point is ideally explored in phase 1 and phase 2 of the study. And as I mentioned before, the assay should be locked down and the cut point pre-specified. If the cut point is modified during the trial, it should definitely not be based on results or unblinding of the outcomes in the clinical trial in which the diagnostic device is being validated. So the validation of the device should not be based on subgroup exploration within the study in which it's being validated. So independent test and validation sample sets are essential for cut point validation. And combining of data from test and validation cohorts, saying that I have a cohort A in which I explored the cut point and I'm validating this in cohort B within the same clinical trial, you cannot establish outcomes based on combining cohort A and cohort B. That's an example of how you don't combine data to validate the performance of the device. And if exploration of multiple cutoffs such that subjects are stratified based on multiple cutoffs, and I'll give you an example in my next slide. It is possible to do that, especially in all-comer studies, if test results are well-defined, and stratification factors specify that all these test results will be used. However, one needs to keep in mind that if there are multiple cutoffs being explored, analytical validation should be performed around each cutoff. One of the biggest examples that I can give from approved diagnostic devices, complementary, is the 28-8 assay that's complementary for nivolumab in non-squamous non-small cell lung cancer. In this clinical study, the efficacy of the drug was measured in patient subgroups who had PD-L1 less than 1%, PD-L1 greater than 1%, PD-L1 less than 5%, greater than 5%, less than 10%, and greater than 10%. However, the clinical trial stratified subjects as they enrolled into the study such that the two arms had even representation of all of PD-L1 expression at each level, and there were no imbalances. And the analytical validation that went with this validated all three cut points for the device. So, and next I'd like to touch upon something that is very specific to the PD-L1 IHC test, and that is the complexity of scoring. And this is a unique issue for IHC test results, and how positivity is defined, or the clinical cutoff is defined, and the implications of what the clinical cutoff is when it's varied for the same analyte across multiple drugs. And as you can see in this table here, when you concentrate on the clinical cutoff and the cell type scored, you see that for Opdivo, in almost all cases, tumor cells are scored, and a score of greater than 1, 5, or 10% is considered PD-L1 positive for lung indication, whereas for melanoma, a greater than 1% is considered PD-L1 positive, and it's a dichotomous and not a continuous score, unlike for lung. So within a single drug-device pair, there are two different definitions of PD-L1 positivity. When you move to another drug-device pair and Keytruda 22C3 in this case, in the first-line lung, greater than 50% tumor proportion scores, or tumor cells being scored as positive, means you're PD-L1 positive. And if you're not a first-line subject, but you've had more than 1 line of treatment, then your PD-L1 positivity level drops to greater than 1%. And this is a lot of information for pathologists and community practitioners to hold in their head in terms of how PD-L1 positivity is described. But the added complexity in the PD-L1 field comes from the use of or incorporation of the immune cells into the scoring system. And when immune cells are incorporated into the scoring system, it's done in many different ways. And for IC scoring by the Ventana SP142 and SP263 assays, it is defined by the percentage of tumor area occupied by PD-L1 positive immune cells. Whereas for SP263, it's a two-step evaluation. First, the amount of tumor area that's occupied by infiltrating immune cells is assessed, and the percentage of that percentage immune input rate occupied cells that are PD-L1 positive. And these are percent areas, mind you. These are not percentage of immune cells or percentage of tumor cells that are positive. So there is a lot of granularity and differences in how these things are scored. And given that these assays identify the same analyte, it could be easy to think that these assays are equivalent and that a lung cancer assessed with any one of these 4 assays is going to give you the same answer. But the clinical validation for these tests have been done at very well-defined scores, and that means that the clinical outcomes that are being reported in the labels apply only to the scores as defined by the diagnostic drug maker with the test of interest. And how these map are still an area of active research at this point. And I want to point out that for Keytruda, while the lung indications use tumor proportion scores, the gastric indication uses a combined proportion score, which takes into account all PD-L1 positive tumor cells and immune cells as a proportion of all tumor cells multiplied by 100. So each system carries its own scoring differences that in turn impact. I've gone over the slide in great detail before, so I'm going to skip over that. So when we have seen multiple applications using different definitions of positivity for a single analyte, what are the issues that FDA faces in assessing the safety and effectiveness of a device? We do not make any assessment of the comparability of the 4 devices. Each device is approved as a standalone and how it performs in association with the drug with which it was fed in a clinical trial, and that is the extent of it. For us, the review challenges remain in how the PD-L1 positivity is described, whether there is implementability, whether the scores can be implemented in the community with safety. That is, will pathologists in the community be able to replicate the scoring that was implemented during the clinical trial? And is it reproducible? And are they all measuring the same biology? Are questions that we ponder out as we review each of these applications. And if the analytical performance is marginal, then one of the things we need to think about is how the clinical cut point and its definition is contributing to poor analytical performance. And we are also needed to assess if clinical cut point can be effectively trained within the community for wider use. So among the other things that impact clinical assessment is the specimens and the variability in specimen or pre-analyticals that might impact clinical outcomes. And here I flagged the specimen data that we typically request all device manufacturers to obtain. We ask them to keep a record of the type of specimen deposited into the study, whether it's archival or it's a freshly obtained biopsy. And in a recent review of the 22C3 test for gastric cancer, it was determined that archival tissue, the PD-L1 was relatively unstable in archival tissue when compared to newly obtained biopsies, which triggered a limitation statement requesting that if an archival PD-L1 specimen is tested for patients in the clinic, and you return a negative result, it might be worth exploring if a second biopsy is feasible because the test result is not logged down because of instability of the antigen. We asked for information on the biopsy location, or the specimen location, whether it was from a primary site or a metastatic site, whether it was a resection specimen or a biopsy specimen. Because the way these specimens are handled might impact what the type of results that they get out, because biopsy specimens are handled very differently than resection specimens. And the pre-analytical variability can result in a different test result. So we request that all this data be collected and included within the PMA application to make an assessment of the factors that might impact test results and outcomes. So what are the analytical validation studies? Having talked all this while about how clinical validation is performed, at this time I'd like to switch gears and go to analytical validation. And what are the analytical validation studies that are required? There's a bit of a lag in the slide loading. Analytical validation is So analytical validation, and here I'm listing all the factors that are important for an IHC test result. And here, analytical validation includes the characterization of the assay in the laboratory before it moves into a clinical trial. So the biochemical characterization of the antibody, so that assay sensitivity and specificity, given that IHC tests use an antibody, the specificity of the antibody is determined. The precision, including reader precision, is determined. So the precision is just retesting over multiple days using different lots between different laboratories are precise, that you get results that are reasonably similar. What happens when you test them across sites? Remember, these are distributed kits. So what happens when they're distributed and if they're tested at multiple sites? That's another important piece of analytical verification that we routinely ask for. Assay robustness tests for the limits of the assay, the boundaries within which the assay performs precisely. Say for a PCR reaction, it could be the annealing temperature being moved around, and for an IHC test, it would be the target retrieval temperature, and the time, and the pH of the buffer being used. If it were to vary by some amount around its ideal, what are the impacts? Do you retain the results? Do you not retain the results? Where are the failure points? Also, within the analytical variables, assessment of pre-analytics, how the sample handling impacts, the presence of impurities impacts test results is important to assess. Also, understanding tumor heterogeneity, whether it's within the tumor or primary versus metastatic, gives an understanding of the type of specimens that would give us the most reliable result within the clinical setting. The stability of the reagents of the specimen also needs to be defined so that the entire test system is defined and its performance within a window of time is well known. And if any controls are provided within the test system, those need to be validated as well. So what are the factors one takes into account during analytical performance assessments? It was, you start from the test. The test is specimen to result. So every step along the way needs to be validated. The instrument, the software, the specimen type, the pre-analytical processing that goes into it. So the pre-analytical processing, validation should be performed with intended specimen type. Suppose the study is being conducted in non-small cell lung cancer. The specimen type with which the analytical validation is performed should be from non-small cell lung cancer. And for molecular assays, sometimes contrived specimens for challenging specimen types can be used. And if contrived specimens are used, what are the functional characteristics of the contrived specimens? It should be well defined. And all studies should follow protocol in labeling. So the label should be locked down much before any analytical, the protocol for the test should be locked down much before any analytical study is initiated. And studies should demonstrate robustness at clinical cutoff. So I talked about many different tests with many different cutoffs, and at each of them, the assay needs to show that for specimens that exist around the cut point, the study is, that the performance is robust, and that there are few false positive and false negatives, and the overall performance meets certain acceptance criteria. So assay validation can be thought of as a matrix. You need to think of each component, and each of them need to be validated. And here I've opened it up to all types of assays and all types of specimens. So what you do is, if it's a PCR using whole blood or plasma, then the assay, the instrument, so it's a PCR assay with whole blood plasma, and you're testing DNA, so the DNA, and the outputs, whether it's qualitative, whether it's mutation in a particular gene. So when you develop this matrix, it immediately becomes obvious the types of things that one needs to do to validate the assay. And for molecular assays, additional metrics need to be established, such as limit of blank, limit of detection, limit of quantitation, specificity or exclusivity, and accuracy against a valid comparator or a reference method. And guard banding and robustness map to IHCs and molecular tests very well. And more recently, with a lot of NGS tests coming online for approvals, the approach we have taken to analytical validation is that for any test that detects multiple genetic changes or multiple mutations, say multiple mutations within a gene, each of the type of genes should be analytically validated, and you should ensure that they can be detected. And multiplex assays such as NGS require complex validation. And if you look at more recent approvals of the Foundation 1 follow-on companion diagnostic, you will see that the validation approach takes into account representative variations of gene sequencing tests. And again, reiterating that analytical validation should take into account specimen and should be done with the final intended use specimen. So for the final leg of my talk, and I think I'm running out of time, so I will move through this a little faster than I intended to. One needs to keep in mind that companion diagnostics started as a single analyte, single indication with a single drug. And the first example of that is the HER2 as the analyte with Herceptin and Herceptin in breast cancer. However, as time has gone by and targeted therapies have taken off, every possible type of analyte indication and drug has come through to the FDA for approval. And I will not go over each particular case, which you might be able to go through this individually. From one analyte with many tests, we have gone to multiple tests for multiple genes with a single drug. We've gone to multiple analytes and a single test for multiple drugs. And then each of these cases have presented us with unique challenges, and FDA has tried to be flexible and nimble in addressing concerns of safety and effectiveness without hampering or coming in the way of development of these tests or slowing down access in the market. And one of the one of the things that I'd like to point out is the follow-on companion diagnostic. A follow-on companion diagnostic is a version 2 or a version of or a test that was not used in the registrational trial for the drug. The follow-on companion diagnostic should consistently and accurately select the same intended use patient population because it was not tested in the clinical trial. What it needs to determine is that it can identify the same population that the originally approved companion diagnostic can do. So one of the ways that one can do that is either testing the clinical trial specimens from the original study with the new test, which establishes that it identifies the same population from the originally approved companion diagnostic device. And this was how COBA's KRAS mutation test was approved as a follow-on. However, it could also be performed with a bridging study, which is a method comparison, which says that we've taken the intent to test population, all the patients who would be eligible for testing, when we test them with the old companion diagnostic and our version of the new companion diagnostic for which we are seeking a claim, they identify the same population. Whereas it's an analytical method comparison, a procured clinical sample set should should confirm that it is the targeted population for which the drug is meant. And when there are discordances, one needs a plan ahead of time on how the impact of discordance will be determined. And also take into account if these discordances are happening because of biases in how the testing is done, or how does these discordances impact device performance. So we need to determine if it's random or there is a bias in the discordance. Are all the test samples that are testing positive with the new device where tested negative would be a clear indication of bias. So having talked about it, I want to switch gears and talk about the other challenges we have for approving diagnostic tests and how we have tried to make things easier. As you've seen with the PD-L1 diagnostic devices, each of these tests have claims and multiple indications, and we decided that the least burdensome approach for clinical validation would still be the contemporaneous review of CDR and CDRH for any given clinical indication. So for when the device came in for urothelial bladder cancer, it was still reviewed by CDR and CDRH, and it still came from a clinical trial. But the analytical validation data for the 28-8 assay was done such that you could leverage data from prior indications. The analytical validation should not change. The precision of a device would hopefully not change just because indications are changed, so long as the same analyte is being detected. So antibody specificity would not change. The tour of tumor, tour of body studies that are part of the sensitivity and specificity are not varied across studies. So or the real-time stability, or this real-time stability in this case is the stability of the reagents that are being used to test various indications. If one were stable for lung cancer, there is little reason to believe it would not be stable for breast cancer or melanoma specimens. And and the pre-analytical variables, given that the analyte is in the same matrix, if it's an FFPE, then you would imagine that nothing should change and you shouldn't have to repeat those studies. So when you have the same device being applied to new tumor indications, you could reduce the amount of analytical validation that's being performed and leverage data from indications with prior approval. And towards the conclusion, all I would like to say is that this is an exciting field. Biomarkers in immunotherapy are very hot, and everybody is excited to explore and research in this area. And the exploration is taking the form of many different tests. Gene expression profiling for immune signatures is often talked about. And last May, we had the approval of Keytruda for a tissue agnostic condition for mismatch repair deficient or microsatellite instability high tumors. So any tumor that is mismatch repair deficient or microsatellite unstable, patient with a tumor with that status would be eligible for treatment with Keytruda after failure of other therapies. So these are new and emerging biomarkers. And another one that's often discussed is the tumor mutation burden by NGS. And here, there are many open questions. And as I said, this is still in the biomarker stage where you could determine tumor mutation burden either by doing whole exome sequencing or doing gene panels. And if one by doing gene panels, what is the choice of gene panels? Are all genes that you're testing in your panel equivalent for mutation burden? Are some more favored than not? And what is the definition of the cutoff? So we see the same issues that came up with IHC coming up here on how you define positivity, how you choose your cutoff, what are the underlying measurements that you need to make to arrive at the right answer. So each of these biomarkers present us with similar challenges as we go forward. So in conclusion, what I would like to say is that for a successful conversion of a predictive biomarker to a companion diagnostic, it's useful to think about establishing biomarker testing strategy very early in clinical development. And if you collaborate early on with FDA and you bring to both the therapeutic center and the diagnostic center of the existence of a biomarker and a test associated with it, one could think about all the possible ways in which to mitigate the risk from developing a diagnostic test because of which a drug trial might fail, or a diagnostic test that is poorly designed that might fail in itself. So using the pre-submission process was critical for new development programs and companion diagnostic programs. And the sooner you interact, the better shape one is into developing a successful program. The last two slides give you links to useful resources. All companion diagnostics that are approved by FDA is listed at the website, as I've flagged here. And below here are all the guidances that FDA reviewers use for review of companion diagnostic devices. With that, I'd like to thank you for your time and take any questions that you might have. Thank you.
Thank you, Dr. Janaki Veeraraghavan, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your questions into the box that appears on your screen and click on the Send button. We'll answer as many of your questions as we have time for. Okay, it looks like we have time for one question. Our question is, when during the review does the companion versus complementary status of a diagnostics made?
Interesting question, and a useful one to be aware of. If it is an a targeted population trial where the drug, the intent to treat population is also already biomarker selected, so only biomarker positive subjects are enrolled into the trial, or only biomarker negative subjects are enrolled into the trial. So if it is a marker selected trial, it ties you into a companion diagnostic paradigm where the test becomes essential to identify the intent to treat population. Whereas if it is an all-comers trial, what could happen is that during the course of review, where CDR and CDRH are contemporaneously reviewing, brings up questions. So this happens during the review process for the drug and the device. As I said, both the applications are always reviewed in parallel. And when they're being reviewed in parallel, the Center for Device and the Center for Drug talk to each other and determine the safety and efficacy of both. The drug review from CDR takes into account the safety and efficacy of the drug, and we review the safety and effectiveness of the device, and in agreement with each other, make a decision, and this usually during the review process. I hope that answers.
Thank you for that answer. Any questions not answered will be addressed via email. I would like to once again thank Dr. Janaki Veeraraghavan for her presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.